Difference between wisdom and knowledge. Wisdom is using what you've learned, not simply just learning something. It might be kind of hard to see because some of it's obscured by the metal railing. But these questions are exactly the same question, just a different aspect of the same thing. Actually, you need to sort of get, you remember what a DNA glycosylase does, which 83% of you did. You just need to apply that to a picture down here. And they got even easier because at the bottom, the answers told you what each of the things were, what A was, what B was. So you really should be able to pick out the wrong or the right answer in this case. The question I put forward to the audience is it so different? Yes, it is a different question, but is it so different that there's 50% success rate difference?
What was the answer for that one? The bottom one?
For the DNA glycosylase, we want to remove a damaged or wrong or incorrect nucleotide, and there should not be any U's in DNA.
Okay.
So the U should be removed. Remember, DNA glycosylases remove a base, but leave the sugar phosphate backbone. So there really should have been two possibilities for things we could do. Remove the G base or remove the U base. Hopefully we should appreciate that the U is not supposed to be there, but the G is supposed to be there because it's the DNA and one's RNA. So the other two are showing you nucleotides, and by adding in the description that D is the guanine nucleotides, which will give you a hint. But here we have the sugar phosphate thing, and here we just have the base part. We see the same sort of trend with the other question, although it's a little bit better. For whatever reason, I don't know, maybe people just don't understand the AP endonuclease better. A lot of people got DNA glycosylase and AP endonuclease activities mixed up. Again, if you understand what it does, it's going to be able to translate that into a little bit more practical application type of thing. You don't ever see this on the exam, but it is kind of fun to put it on quizzes every once in a while. But I mean every semester, to see how students do on let's just learn the fact versus let's apply information to a drawing. I don't know the last time you saw a hand-drawn DNA structure. Maybe that's the problem. My artistic skills are lacking completely. Who says that's the problem? Dr. Richard can't draw. That's why I didn't know what was going on. Anybody? You guys all missed a fun talk, a great yesterday. Should all go. Why didn't you tell everybody? Why didn't I tell everybody? I forgot to tell everybody.
I was trying to use a few people in the room.
You
The biology department.
You didn't need students there. Well, that's really what the target audience was. He was giving a talk on his internship. Which was cool. I had a question, but I didn't just ask. I had to go deeper. So I'll ask you right now. If the company is so interested in keeping duck populations high, do they ever go around and collect the eggs from a nest that's lost its egg and hatch those ducks and then put them out?
Yeah, they do have hatching programs, but it's very inefficient. It's better to let them do naturally managed programs.
So if you want to know more, then what's your appetite for ducks? Brings the guy. Or if you want to go outside, hear what he did for an internship. And then you can do it. Not so. Okay, yeah, go ahead. Sorry.
So
I was going to start.
there was a question said, what is the name of the mechanism that causes dynamic mutations to occur? The correct answer was replication slippage. I just wrote slippage. And I didn't get
I see a lot of people slipping outside. It's not causing dynamic mutations. So that is the only fallback I have. The other fallback is it was only worth 1 point. I don't like to give half-point partial credit on quizzes. It's more important for me. So it's more of an incomplete answer because it's not a complete answer. It was kind of wrong. Just saying slippage is not enough because when did the slippage occur was not in the question or in the answer, which is why replication slippage was needed together. Right? Say when during replication do dynamic mutations happen? Read it again.
It says, what is the name of the mechanism that causes dynamic mutations to occur?
Yeah, so So replication slippage is the correct answer. Slippage is the incomplete answer. Any questions about the quiz? Or you also ask, are there any questions about the quiz? Okay then. Our next quiz will be posted either later today or tomorrow, which means due on Monday morning, 9 a.m. Yay, 9 a.m. Monday morning. Then we have another quiz after that, which will be due on 9 a.m. on Monday morning. Next Thanksgiving. That's the test. That's the final. We're done. But we don't do a whole lot in this class, is kind of look at the big picture, so I thought it was worthwhile to sort of take a step back for a second. And if you think about, well, why do we really need to care about regulation in the world of GEX? I've tried to give you one answer already for why we should care about this, is because regulation controls when things happen. But in this class, mostly we talk about genetics. It's the idea of connecting a genotype and a phenotype together. So let's think back to the beginning of the semester. Who remembers what a simple trait is? Simple trait versus complex trait. Only one gene. Simple traits. Complex traits are controlled by multiple genes or environmental factors. And so in a simplistic view, which Gregor Mendel gave us, we have purple or white, and there wasn't anything in the middle. We sort of know there's other things going on. We can have a gain of function mutation. So let's say there's a gene, gene A. And we make a missense mutation that increases its activity. So normally it's 100%. But if we mutate it and we get a gain of function where now we're at 200%, this could produce to be a problem. There are a lot of diseases that are associated with too much of a certain protein or a certain metabolic process going on. So that might make a disease. That's what we think about the function or the activity of a protein. As we increase it, we could create a disease state. The reverse is also true. If we see a loss of function mutation, that could also lead to a disease, and it most likely probably a different disease. So changes in the DNA sequence of the gene affect protein function, which could lead to an altered physiology, which could ultimately lead to a disease state showing up. We also have hyper and hypomorphic effects, so we can have things that will affect expression. So sometimes we may get something where we get more expression. And so instead of getting the normal expression of 100%, they get 2 times as much of a protein present. Ultimately, probably gonna lead to the same thing, disease state. There's too much of that protein around, so we get the same sort of disease. And likewise, if we go the opposite direction, if we decrease expression and the activity is still 100%, then we're sort of getting in the same state where we have half as much we need, which could lead to a disease state. But still, it's only one gene. What if we mutate this other protein over here? Gene B. Now gene B is a regulator of gene A. So gene A, normally 100%. If we mutate it and make it 200%, how does it affect the expression of gene A? If it's 200% more effective, yeah, it probably increases. Well, it depends. Is this a repressor or an inducer or an activator? We don't know because it doesn't matter, but you're getting the idea is that in a world of regulatory proteins, it's not just changing the gene that does something, it's the gene that controls that gene that could be the problem. Keep going back. There's probably a gene C that controls gene B, which controls gene A. So you start to see how things get a little complicated after a while. But you don't need to actually break the end gene. You can break something in the beginning of this whole pathway of connectivity that can all end up leading to the exact same spot. Which is why a lot of diseases in humans are really hard to sort of identify the cause. Because you break any number of things along this chain and they all end up giving sort of the same result at the end of the day. Given enough time, sometimes you see things called compensatory mutations. So if there's a mutation that causes this to increase, maybe down the line, the expression gets lowered. So another mutation later on brings this down so it's about normal again. And given enough time, sometimes these will occur where you don't really change this, you try to bring it back to this, you change something else. Sort of again, dial these things back. You do see these sort of keep going on, you break one thing, you alter something to fix it. So you do have some problems with how things are connected together. Again, complex traits are really hard to deal with because you never know exactly what is the true cause of something. This is why understanding regulations helps us sort of think about genetics. Anything that controls expression or affects expression can have a pronounced effect on the phenotype that we see. Things covered today will be on our quiz 8. Which will finish off a lot of the things we've already talked about. So ways that we can control regulation. Ways we can control gene expression using regulation. So far, all we've seen are ways to control transcription. Either by using DNA binding proteins to affect RNA polymerase's ability to bind to the promoter. You've seen attenuation, which is using translation as a proxy for amino acid content. Decided we need to make more of that amino acid. Rabbit switches are the weird sort of thing we don't talk a lot about, but altered RNA structures control transcription. Long non-coding RNAs, decreasing gene expression of entire chromosomes, not just specific things. In induction, activation, repression are typically only affecting one thing. One gene is controlled by one protein. Attenuation is controlling an operon, but still an operon for one thing. Typically with the long non-coding RNAs, that's when we see a lot of things being controlled. Because again, we can sort of knock out the entire X chromosome by using the X RNA. I'm not saying all long non-coding RNAs work the same way, but does allow us the ability to control a lot of genes at once. Same thing is true for methylation. Methylation is not just doing it one at a time, it's controlling a lot of genes in a lot of different places. Now, if we go back to the idea of repressors, inducers, and activators, we have something that is kind of like that. This is something that is eukaryotic. In eukaryotic organisms, there are pieces of DNA that can control transcription. And so in bacteria, we have these proteins that bind to different DNA sites to control transcription because they affect the promoter. But in a eukaryotic organism, enhancers and silencers are different DNA sequences. So as an example, the enhancer could be here. Or this could be the silencer. And the enhancer or silencer DNA sequence could be anywhere. Sometimes they're actually in the gene coding region. It's always a strange sort of thing to think about. But one of the most important things about these is they are not proteins. Enhancers and silencers are not proteins. They're DNA regions, DNA sequences that proteins bind to affect transcription. In the presence of these, either stimulate transcription for an enhancer or inhibit transcription if they're silencers. These enhancers and silencers are organized into regions. At least the chromosome is organized into regions to control whether these enhancers and silencers work where they're not supposed to work. And so couple this with the next thing because they do go together. The chromosome is divided into parts. We call these domains. The technical term is topologically associated domains, or TADs. You know a TAD? Usually TAD's not a very common name in Tennessee from what I've heard or seen so far. The TAD is a DNA region, and it's a pretty large DNA region. The importance of TADs is kind of like bookmarks or bookends. They control a certain region of the road, and the road being the chromosome. And specifically for us, one of the cool things about TADs is they prevent silencers and enhancers from affecting things that are not in that TAD region. So if we take a little bit more expanded view of a chromosome, and at the ends are the telomeres. Somewhere is the centromere. See how well we remember things. What is this chromosome called based on its centromere position?
Is it in the middle or towards the left?
It is not directly in the middle. It is not metacentric, because that is in the middle, but submetacentric, yes, would be the answer. If it were close over here, it would be acrocentric. But if we take this chromosome and we break it into parts, which is kind of what is done through the chromosome, each of these regions is a TAD. So called TAD1, TAD2, whatever you want to call it. And there's enhancers and silencers in TAD1 that are going to affect the genes in this region. You don't want this region to affect other regions of the chromosome, because then you're getting things doing things you're not supposed to be doing. Everything in the nucleus is all jumbled on top of each other. That's the other reason why these things are really important, is the DNA is wrapped around these histone proteins and wrapped around each other, then wrapped around each other, then squished into the nucleus. A lot of DNA sequences are next to each other when they're not necessarily next to each other. Chromosome 2 could be right next to chromosome 12. An enhancer from chromosome 2 could affect chromosome 12 genes if we're not careful. And that's why these TADs are really important. To try to localize enhancers and silencers to specific positions so they only affect the genes they're supposed to affect. And like the slide says, this organization is tissue specific, so we're going to gene regulate things so that certain things are turned on in liver cells that aren't turned on during heart cells, and vice versa. Not a whole lot to this idea, but again, still, we see this similar type of thing that we talked about earlier. The things that are transcribed more, the things, parts of the DNA that are highly expressed, the parts of the DNA with the most genes, typically go to the center of the nucleus. Things we don't need a whole lot, or on the outsides of the nucleus. And so the nucleus is pretty cool. It's really neat organization potential. But still, TADs, working in conjunction with silencers and enhancers, are forms of transcriptional control. Next, we have a form of translational control. This is not going to affect the ability to make an RNA, it's going to affect the RNA specifically. And so in RNA interference, we are trying to target messenger RNAs for destruction. Messenger RNA is gone, no translation of that RNA happens, which again is going to help us control gene expression. We're not stopping the first step, we're stopping the second step. As you see, it's widespread in eukaryotic organisms, not very common in prokaryotic organisms. But RNA interference. And it works on a very simplistic process. Double-stranded RNA molecules are considered to be bad, because maybe that's viral DNA. And let's kill viral DNA. But again, we are trying to destroy a messenger RNA using a really small RNA. And this really small RNA goes by two names. Really small RNA or microRNA. Which one's bigger? A really small RNA or a microRNA?
Really small.
They're both the same size. It's a trick question. The only difference between them is the difference that we don't necessarily care about. Sort of where do they come from? But what you care about, the siRNA versus the miRNA, is what they are typically used for. And as you can see, the miRNA has multiple targets or less of a specificity, which gives it multiple targets, whereas the siRNA is more specific, giving it more or less a single target. And so while S means small and M means micro, for us it's better to think about M being mini, not M-I-N-E, but M-A-N-Y, many as in lots or multiple. Many, the mRNA, miRNA, does lots of different things. Whereas the S stands for specific, single, it only affects one thing. Now how they work is not really important, but they bind to their target RNA, and that triggers a digestion process. The enzyme that's involved in making the miRNA and the siRNA is called dicer. A lot of students confuse dicer as the thing that's going to destroy the messenger RNA, but it's not. It's the thing that makes the siRNA and the miRNAs. Where do these come from? Something we're not really going to talk about. But people have been studying this for a while, and there's a few potential sources for these things. Most of them are, again, degradation products of other RNAs. So, can we think of an RNA that we make but don't do anything with? Let's think about this for a second. We're talking about eukaryotic organisms. Let's think back to things that are transcribed versus things that are translated. Yes, the introns are what you think about. Introns are not used for anything, at least as far as translation is concerned, but maybe they could be used for this. But is that likely? Is the intron sequence in the messenger RNA that is used to translate? No, because we get rid of the intron sequences. So we need something that's going to match the exon, or one of the exons if we're going to base pair it to the mRNA to use it as a target. So it can't be the intron sequence. It has to be something else. They're on the right track. There are RNAs, or the RNA that we make, not all of it's used to make the primary, or what we call the mature RNA. Some of it might get used to make these little small things that get used to control transcription, or control translation, by causing the mRNA to be degraded. They've been used for a lot of different purposes. Here's a story about trying to use RNA interference to prevent mosquitoes from reproducing. If you can prevent mosquitoes from reproducing, you can potentially prevent the spread of mosquito-borne diseases like malaria, and plus we have Zika, chikungunya. What's another good mosquito disease, you know? Let's go with those two. I talked to Gribble about this. Well, not maybe this, but about mosquitoes, if you're careful with mosquitoes. Is it being a base item for Gribble's medical and veterinary entomology class? You probably hear about mosquitoes and the diseases they cause. Actually, probably guarantee it. But those are insects. Who cares about insects? Let's deal with cancer. Yeah, cancer. Everybody loves cancer, right? Who's pro-cancer? No? It's a funny joke. It's a sad joke. But a lot of students enjoy hearing about cancer. It's kind of a fun topic. Not fun, but interesting topic. I use the words or something that's bad. Hey, did you use microRNAs to try to target cancers? Does it work? I don't know, I didn't read the article. But this suffers from the same sort of problems that CRISPR suffers from. How do you get it in? And how do you ensure that it gets to all the cells that it needs to get to? But it's not just cancer that RNA interference is being used against. There are lots of diseases that you can see on this list. A lot of them are cancers or things that are related to cancer. But this is a formatting problem. It's chronic hepatitis B, not atherosclerotic cardiovascular disease hepatitis B. It's the chronic goes with hepatitis B. I was like, what? It's weird about this. This is weird. Anyways. Oh, here's something about long non-coding RNAs. Ask me why this is not next to the long non-coding RNA slide. I don't know. Let's fix it. You know it's gonna drive me crazy. Are we there? Are we there? We're not there yet. Who likes it when I go off topic? Anybody? Hey, let's start to learn. Yay. Let's learn what OCD initiatives. It's okay, everybody can pause on OCD. Think so, talk to Dr. Fitz. He's OCD. Rearranges the room all the time. Me, I just complain about it. Seriously, I should be doing other things than doing this. I can do this after class. Am I Sheldon or am I Monk? Does anybody know Monk?
Mm-hmm.
Does anybody watch the movie? TV show?
The TV show was great.
Okay. Let's save it. That's my other problem. Make changes, but then I don't save them, and they don't get actually fixed. Now the real question is, I made several mistakes on our quiz. Have I fixed them? Or will I not remember to fix them until after those questions get used again next semester? Like, oh shit, I forgot to change that. But getting back to the real point of the story, RNA interference is used for a lot of different things and is a way to control translation, but not transcription. What does this say? It turns out that sometimes you can do some weird types of modifications of RNAs. I can't remember the exact example, it's a muscle protein. A single gene sequence is used to make two different types of muscle proteins depending on which introns get removed and which exons get glued back together. And so sometimes genes that have multiple products because of the exons that are used are also regulated differently. And it's just something to kind of think about. This is not what that slide's about, but it is about the tail. The tail is actually the most important thing about the messenger RNA when it comes to a regulatory mechanism. The cap, the 5' end, is not going to do anything for regulation, but the tail is pretty cool. The tail is kind of like a ticking clock for the messenger RNA. As the tail gets shorter, the lifetime of the messenger RNA gets less. What you want to think about is why would cells want to control how long the messenger RNA stays around? That's what this basically is. How can we control how long a messenger RNA sticks around? Do they have any thoughts on why we want to control how long an RNA stays inside the cell?
Can it be used like twice?
It can be used more than once.
But I would say like.
It can be used 100 times or 1,000 times. And that's the issue. Even if we have a protein that binds the DNA to stop transcription, maybe it's already made 100 or 50 or 1,000 RNAs already. And what do we do with those things? Those things are still out there and we can still be making that protein. So what we see is transcriptional control mechanisms coupled with translational control mechanisms to sort of get us back to where we want to be really quickly without doing a lot of things. And this is only a eukaryotic thing, but in bacteria, mRNAs have what's called half-life. So they only stay around for a certain period of time. The cell wants to make more of that same protein, has to keep making RNAs. It's just a way to sort of control, sort of a cell control mechanism. The RNAs, you make them only last for a minute or 5 minutes, then that's a way to control how much that protein is made from. But as we degrade our tail, the mRNA gets useless. And I have think about this in the same kind of sense we talked about telomeres. Remember, telomeres get shorter, and sort of as they get too short, cell division stops. As these tails of the mRNA get too short, translation is not going to be possible anymore. How it happens is not important, but the shrinking of the shell, shrinking of the tail, stops the translation of that mRNA. And there's two different ways to do it, but it doesn't really matter that they're two different ways. Just that shortening the tail means the mRNA is not translated anymore. You don't have to degrade the mRNA completely, you just have to make the tail too short. Now the other question I have for you is, do you actually appreciate seeing these abstracts in the notes every once in a while? Is your question easy?
Yeah, wait, so we don't need to know those pathways?
These two things? No. They're both the same, really. The tail is made out of adenines, which is why they're called deadenylations. It's really not important to differentiate them. We're just trying to shorten the tail. Less useless sort of knowledge. It is a translational control, which is always important to think about. Very few translational controls, many more transcriptional controls. I know this is about microRNAs and how microRNAs can be involved in controlling certain types of cancers. I started talking about this idea, but sort of stopped myself. There are genes in eukaryotic chromosomes that do make two different proteins, which is a weird thing because typically in the world of molecular biology, we say one gene makes one protein, and that's all it is. But it turns out that in eukaryotic organisms, sometimes they double up. Depending on which exons get used, you get one protein. I think it's like smooth muscle versus, what's the other smooth muscle? It's not smooth muscle, you know? It's like an AMP. Not cardiac muscle, skeletal muscle maybe. It's like skeletal muscle is some set of exons, and then smooth muscle is some set, but they use some in conjunction. That's what we call alternative splicing. So which exons you use controls what protein you use or protein make. But it turns out that when you have this alternative splicing possibility, there's differential regulation that happens as well. Not really anything to worry about, because again, it's just an example of research sort of catching up to lecture or vice versa. The last thing we do have in this section are ways to sort of study gene expression, which we talked about one already. So you may remember the one example of a method to study gene regulation and talking about replication and the things that came from replication.
PCR?
Can you be more specific? Because we actually have 3 different types of PCRs.
Reverse transcriptase PCR and quantitative PCR.
Not reverse transcriptase PCR specifically. qPCR is the technique that is used to measure gene expression. If you combine qPCR with RT-PCR, you can measure gene expression of RNAs, but just doing qPCR is what you think about. So qPCRs are ways to study gene expression. We also have microarrays, which is basically a qPCR on overdrive, because you create a lot of things at once. And a very simple or original thing was called the Northern blot. These are ways that we can measure RNAs. By measuring RNAs, we have an idea of how much gene expression is happening. There are also ways to measure proteins, and one easy way to measure or look for specific proteins being present is called a Western blot. And I'm not going to talk about them a whole lot, other than just what they do. But in a Northern blot, we're looking for a specific RNA molecule. We're basically running a gel and then detecting the RNA by using antibodies or a DNA RNA probe. For Northern blots, you can only study one gene at a time. That's why it has limitations. So after a while, somebody had the crazy idea, let's sort of change this, which is where we got the microarray. It's called microarray because it's done on a microscope slide. Not exactly a microscope slide, but something that looks like a microscope slide. And here's sort of a picture of it. The slide is pre-made by a company to contain every transcript that an organism could make. You have to buy a slide for a certain organism. And you grow that organism and you're trying to study under one condition and then a second condition and see what happens. In this case here, they're looking at a healthy cell gene expression pattern versus a disease cell gene expression pattern and look for the differences. Because typically where the differences are is probably what is causing the disease state to show up. And by looking at the patterns over here, you can say that some of them are not turned on, some of them are turned on one condition but not the other, and vice versa. So this particular thing, the red ones, are the things that are problematic, because they're not normal. And we can identify what gene this came from, and maybe study that gene and see if that gene is the problem that's causing the disease. Or is it the other spot, which is a different gene? And so you get a lot of information about a lot of genes all at once. So you have to do some tracking down to sort of figure out what is actually the problem. But to be able to study genome expression of an entire cell all at once is a pretty cool thing. It gives you a lot of data very quickly, and you can spend years sort of analyzing the data from just one experiment, is what a lot of people do. But it allows us to study gene expression by looking at RNAs. Rosa blots study gene expression by looking at RNAs. Western blot is a way to detect a protein. So again, we're looking at gene expression in one level, just the translation level rather than the transcription level. And how it's done is not important. The question that you're going to see is how is detected using a Western blot. DNA, RNA, or protein. Or DNA or RNA, or some combination of all things. Same thing with an oral blot. Same thing with qPCR. Same thing with microarrays. What are we detecting? What are they used for? Ways to study gene expression. Now, I'm not quite done with ways to study proteins, because there's one other aspect that is worth talking about. And there's two other types of common experiments that are done that sort of relate to the idea of gene expression. We can ignore the slide because there's nothing really there that's worthwhile. But there's two techniques that are shown here. The first one, called CHIP, is a way that we can identify which proteins bind to which DNA sites. This will allow us to identify the activator of a certain gene, or the repressor of a certain gene, or the aporepressor of a certain gene. So being able to say this protein goes with this DNA sequence allows us to say maybe this protein is regulating this gene in some way or another. We have to do other studies to figure out how the regulation happens or what type of regulation it is. But to be able to say that this DNA binds to this, or this protein binds to this DNA region, tells us something about the protein and what it does. The other one is a little bit weirder. It's called the yeast to hybrid system, because that's where it's developed in yeast organisms. For the yeast to hybrid system, instead of looking at DNA and protein interactions, we're looking at protein-protein interactions. And we haven't really dealt with any protein-protein interactions at all in this class. But if I included a few other examples of eukaryotic regulation, we would need to talk about one protein interacting with another protein to control. And that's what it gives us an idea of what does this thing do by who does it talk to and who does it interact with. The last thing we need to think about is a preview of where we're going in the last little bit of stuff we're going to talk about. Is we're no longer talking about controlling transcription or translation. How can we control proteins that have already been produced? And if we use DNA binding proteins, we stop the RNA from being made. We can degrade the RNA, so we stop the protein from being made. But what do we do with the proteins that have already been translated? Those are potentially still out there doing something we don't want done. And there are a couple different ways that we can approach dealing with those proteins. One is we can modify them to turn them on or off. We can modify them so that we degrade them or leave them alone. Or we can bind proteins to them so they no longer function. Which, if anything, this is where we might see the use of the yeast to hybrid system, is to see which proteins bind to other proteins. Now, it's not the next chapter that this is gonna show up. It's the next information after the next information. And if you look online, you probably noticed that new set of slides are posted. And this is not the important part. The important part is the slides that come after this. But this will set the stage for the things that we're gonna talk about next. Because cancer is all about problems with the cell cycle for the most part. Cells dividing when they're not supposed to divide, which is the key aspect of cancer cells, is uncontrolled growth. Lots of cells being made in a really short period of time, really quickly. It's how we get those tumors forming in people's bodies. But a cell undergoes a certain set of steps to divide. And this cell cycle is different for one cell type versus another cell type. For meiosis, it was all about germ cells making gametes. We go from a diploid cell to a haploid cell. Now with mitosis, we're gonna take a diploid cell called a somatic cell and make another diploid cell, actually 2 diploid cells officially. Cell cycle controls this process of going through the process of copying our DNA and then going through the stages of mitosis and cytokinesis to create 2 new cells. And as we go through some of the things that can get broken, we're gonna deal with things associated with G1 phase and S phase, and things associated with some of the phases of mitosis. I'm not gonna spend a whole lot of time on this right now, because we're gonna pick up a lot of it later. But hopefully you remember something about meiosis, because if you do, it'll make mitosis a lot easier. You just need to forget about a few things. One of the things we don't spend a whole lot of time on with meiosis is this thing we call the interphase. The interphase is very important for mitosis and the normal cell cycle. Germ cells don't do this because they make gametes and that's the it. They're done. But for every other cell in your body, when they reproduce, they're going to reproduce again at some time interval. An interphase is what we call G1, S phase, and G2. The G stands for gaps. They are sort of resting periods, preparation periods, kind of growth periods. The S phase is when we copy our DNA. So we're talking about gene replication. Fun things will have to go on. Does anybody remember the repair mechanisms that are associated with replication? Is that the last exam, or is this going to be exam 3? I don't even remember anymore. I think it was last exam, right? Yeah, right. Mismatch repair, proofreading activity. That was last exam, right? Sorry, it's also in this exam. Just words, not as mechanisms. That's one thing we will look at. Repair mechanisms and how these coalesce into the cell cycle. We are going to draw from a lot of things. Let's just make this sort of the penultimate, not penultimate, the capstone of the class. Take everything we've learned all semester long and feed it into understanding how cancer works. One thing that we have talked about already is chromosomes and chromatids, and hopefully you remember about when chromatids show up and whatnot. I don't know, I'm saying whatnot today. One of the more important things that we have to deal with mitosis and the cell cycle is the presence of checkpoints. What's the purpose of a checkpoint in general? Anybody? Have you ever been stopped by a checkpoint? What are they trying to do? See if you're drunk? Or are you transmitting humans? Or are you bringing fruit illegally into the country? So checkpoints are checking to make sure you're right. The checkpoint for us in the cell cycle are going to ensure that things have been done properly up to the point, so that when we go to the next step, there should be no problems. The checkpoints also allow places where the cell cycle can be paused or stopped if something really bad has been detected. When it comes to cancer, it's not just that these cells divide when they're not supposed to, A lot of them will ignore the normal checkpoints that exist. So they basically, something is broken where they don't obey the rules anymore. And that's something that is sort of interesting to think about in the context of regulation. I really should include a copy of this slide earlier as well with meiosis. Again, when we first saw chromosomes, they weren't viewed as being two different chromatids. It wasn't until later that the two chromatids were able to be visualized. I don't know if you can see the two sort of things here. You can see that demarcation between the two and all these things. Whereas up here, it's just again one big line, but those one big line is two things. Trying to get to a point where we can actually start talking about something fun. Question, do you all remember this from last test at all? Chromosomes versus chromatids? For meiosis, we had to worry about when the homologous chromosomes were separated and when the sister chromatids were separated. We don't have to worry about that for mitosis because there's no synapses, so no crossing over. So we're much simpler process for mitosis. But you may see questions on DNA. Actually, anything from mitosis slides will not be on your quiz, so don't worry about that. It's the end of regulation. These will show up on the next quiz after this quiz this weekend. So we notice when I get to this slide, this is where we're gonna start on Friday. Talk about mitosis, remind you about what are the things that go on. It won't take very long. And we'll dive directly into some of the more complicated parts of the semester. Sorry, the most complicated part of the semester was coming last week. Maybe you thought other stuff earlier was more complicated.